Hi, everyone. My name is Joe Unsworth. I'm a research associate at the Rosenstiel School of Marine and Atmospheric Sciences at the University of Miami. And in this video, I'm excited to share some of the work we've done to improve the efficiency and effectiveness of coral restoration efforts around the world. For anybody unfamiliar, this process is commonly called coral gardening. And what you see here are various examples of coral nurseries where coral is cultivated. You can see that different organizations have kind of put their own spin on these structures, and all of them are made using very inexpensive materials, and they grow coral very fast. So once the coral is grown, fragments are taken down and outplanted onto reefs in order to augment native populations and improve their chances of persisting into the future. So here in Florida, most organizations use one of two methods to attach their corals onto the reefs. We have the nail and cable tie method, which uses a galvanized steel masonry nail in order to kind of hold the coral onto the reef while it provides its own attachment points. And other places use a two-part epoxy putty in order to essentially glue corals directly onto the reef substrate. Both of these methods lead to high growth and survival, but it takes a lot of time in order to outplant each individual coral. So this is kind of the problem that we're trying to overcome now, is that we're growing coral faster than we can outplant it. So in order to overcome this bottleneck, several organizations have begun using cement as an outplanting tool. Cement is one of the most ubiquitous materials on the planet. It's also extremely inexpensive, which makes it ideal for restoration projects, especially remote ones where access to materials and funding is usually limited. It's currently being used by programs in Puerto Rico, Belize, the Seychelles, and throughout Florida. But its use in the context of reef restoration is pretty understudied. So far, nobody has formally tested the impacts of cement on corals or compared it with any of the existing methods. So we need to begin developing best practice guidelines in order to discover the full potential of this new attachment tool. So our first objective in this project was to develop a better coral attachment technique using cement. This is kind of a broad overview of how we approached this, the different tests that we did with different cement mixes in order to eventually settle on one final ideal mix. So in the lab, we tested the physical properties of each mix, such as its cohesiveness and ability to hold corals upright. We also looked at the sediment cloud that each mix produced when used underwater, which is kind of a common concern of using cement for coral restoration. So mixes that produce a large sediment cloud may be harmful to nearby organisms when we're diving on a reef. So once we narrowed down the initial set of mixes, we outplanted corals with each mix to see how each one performed in the field, to see if there were any negative impacts on the corals in terms of survivorship. So we ultimately settled on a cement mix that showed 100% survivorship. Perfect. This is a mixture of 1 part Portland cement to 1/10 of a part microsilica. Microsilica is actually typically used in structures that are constantly in contact with water, such as bridges and dams. So by filling in the gaps between cement particles, it lowers the permeability of the final hardened concrete, and reduces the amount of chemical leaching out of the concrete, as well as making it more resistant to corrosion by salt water. So here's how we use it. So before the field day, we prepare the dry ingredients, and once we're ready to plant in the field, we combine it with a measured amount of salt water. We tested with both fresh and salt water to see if there was a difference. There was not. In terms of survival, coral survival, and consistency, everything was the same. So we decided to use salt water here, because obviously it's much more abundant when you're working out in the ocean. We then funnel it into the same icing bags that a pastry chef would use to ice a cake, and then we secure the tops of those with plastic zip ties. So far, the pastry bags we've been using have been plastic, disposable ones, but we are in the process of trialing reusable canvas piping bags to try to cut down on our plastic waste. Underwater, we remove the tip and squeeze as much cement as we need onto the reef before we add the corals in. This mix holds corals upright, and it hardens in about 90 minutes, which is roughly the length of a typical outplanting dive. So here's how cement compares in terms of cost. This is in terms of cost per coral fragment planted. We can see that cement comes in at about the 10th of what the other methods cost, which is a huge difference when we're talking about outplanting on the scale of tens of thousands of corals. I should mention this is just based on our suppliers, so this may vary based on different locations. We've also tried this method with our Rescue Reef citizen scientists. Participants on expeditions where cement was used were able to outplant more corals than expeditions where nails were used. And we also took post hoc surveys, found that participants generally found outplanting with cement to be easier and more enjoyable. So this is really a huge benefit of using this method, is that it's very friendly to citizen scientists. So in addition to finding the ideal cement technique, we also wanted to use this new cement technique to test new ways to achieve large complex coral colonies in the wild. So here's an example from Fragments of Hope, which is an organization working in Belize, where they used multiple small fragments of coral to plant within a single cement base in a very kind of dense cluster. So as we can see, these outplants show terrific growth. This is after just a couple of years. So we wanted to see if we could replicate this in a branching coral. And this is similar to a concept that's kind of being proven for coral species with massive morphologies. They typically show very slow growth rates, but scientists at Moat have found that by fragmenting these corals into very small pieces and outplanting them in dense configurations, the corals rapidly grow and fuse together to form one single colony. So in the images on the right, the corals were able to completely encrust over this structure in just 1 year. So we want to know if this can be replicated in a branching species, specifically Acropora cervicornis, which is the most commonly restored coral species in the world. So this is the experimental design we used. We had 2 restoration sites across off the shore of Miami, where we outplanted large individuals from 5 different genotypes, all again from the Miami area, and compared those with single genotype dense clusters, again from the same selection of genotypes. These are 7 fragments of roughly the same size, and we tried to keep the total amount of tissue within the dense clusters equal to the total amount of tissue within the large fragments. And then we also have dense clusters consisting of multiple different genotypes to see if that had any effect on growth or survival. So having multiple genotypes in such a close proximity, this might seem like a recipe for intraspecific competition, which is competition between different genotypes among the same species. But this actually closely replicates the genetic landscape of natural staghorn thickets. So a thicket like the one that you're looking at now may have as many as 30 distinct genotypes growing around and between and within one another. So this is kind of what we're going for. We wanted to see if this, if there was any difference between single genotype or mixed genotype. And so far we've monitored these corals after just 1, 4, and 7 months. Here's a graph showing their growth in terms of volume expansion. So a value of 1 here means that the coral has effectively doubled its volume. We can see that not much difference between the 3 treatments. Not only are the dense clusters, single genotype dense clusters, keeping pace with the large fragments, the clusters with multiple genotypes are also keeping pace with the large individuals. So this is kind of a bonus because they have a significant benefit, we think, in terms of increasing successful sexual reproduction. So the ability for corals to sexually reproduce is currently restricted by a lack of synchrony. So successful fertilization is all about having having gametes of 2 different genotypes in the same place at the same time. So a mismatch of just a couple minutes between 2 genotypes can significantly reduce the rate of successful fertilization. So at the finest scale, this is triggered by chemical cues released by the coral. So as a result, corals that are closer together tend to spawn more synchronously than corals that are further apart. For example, if 2 different genotypes are too far apart, they won't spawn together, and neither will be successfully fertilized. So by keeping multiple genotypes close together, we may be able to maximize the number of different genotypes spawning at the same time and increase the likelihood of successful fertilization. This then increases the likelihood that the corals will eventually settle and begin to adapt to changing environmental conditions like rising temperatures, for example. So as a final example from the work that we've done, we recommend that restoration organizations first begin integrating cement into their protocols. We found it to be much cheaper and more effective than either of the two other methods. So there really don't seem to be any downfalls to using this new method. And we also recommend using multiple different genotypes within these dense clusters. Again, the dense clusters are much more preferable in terms of growing. It's much more difficult to grow and transport a single large fragment versus just having a nursery full of medium-sized fragments and constantly pruning off small fragments for use in these dense clusters. And mixing the different genotypes, again, has benefits in terms of increasing successful spawning once these dense clusters form into a sexually reproductive colony or thicket. I now have to acknowledge all of the people who helped make this research possible. My co-authors, Dalton Hesley, Martina D'Alessandro, and Diego Lirman, as well as my cement experts, Chris O'Hare and Prannoy Sereneni, everybody in the Lirman benthic ecology lab at University of Miami for helping with field and lab work. And this project was funded by the Moat Protector Reefs program, as well as grants from Miami-Dade County and the National Oceanic and Atmospheric Administration. Thank you everybody for watching. I look forward to your questions. Again, my name is Joe Unsworth, and you can find my email down at the bottom of that page. Thanks everyone.